Okay, hello everyone. Welcome and thank you for joining us for the repeat viewing of the first in this series of IUCN Red List webinars. These webinars are designed specifically for members of the IUCN Species Survival Commission, so thanks for joining us. Please note that these webinars are being recorded and we will make them more widely available through the website later on in the year. I'm just going to turn off my email notifications as well. Sorry for that. Okay. I'm Caroline Pollock and I'm the Senior Programme Coordinator for the IUCN Red List Unit in Cambridge in the UK. And the main role of the Red List Unit is to manage and update the IUCN Red List of Threatened Species. And part of that role involves providing guidance and support to many of the SSC members and other people preparing and contributing to Red List assessments. So we know that you invest a lot of time and energy on preparing Red List assessments, so we really want to help support you through this process. Every time we do a Red List update, we often see that there are certain areas that people obviously struggle with, so the whole point of these webinars is to help you through various parts of the process. In this first webinar, we're going to focus on the IUCN Red List Criteria. And we have chosen 4 main topics to cover, but we're not restricting everything on those topics. We will be talking about data uncertainty, incorporating uncertainty into assessments, severe fragmentation, calculating generation length, and if we have enough time, talk about climate change and incorporating climate change into assessments as well. But during the registration process, we also asked you to highlight questions and topics that you want to hear about. So we're not going to restrict it to just those 4 topics. We'll also try and answer your questions throughout this webinar. So if you have a question, please do ask using the Q&A function. There's a Q&A icon at the bottom of your screen. We'll field questions throughout the webinar using that. So both myself and Clay Meredith from Albuquerque will be fielding questions as our main speaker is talking. So I'm delighted to welcome our first speaker for the webinars is Professor Resit Akçakaya, the chair of the IUCN SSC Standards and Petitions Committee. And Resit is also one of the main people who was involved in developing the quantitative criteria for the IUCN Red List. So I'm very pleased to welcome Resit, and I'll hand over to you right now. Thanks.
Thank you very much, and welcome everyone. I am Resit Akçakaya, and as Caroline said, I chair the Red List Standards and Petitions Committee. My day job is a professor. I'm a professor at the at Stony Brook University, which is part of the State University of New York system. So I'm coming now. I'm sitting in my home in Long Island, New York. Okay, now I will share. Let's see. Okay. As you were notified, this seminar is about your questions, and especially covering the more complex topics and difficult questions. So based on the questions that were submitted, I selected some and I changed them a bit so that they are more general, and they they also include the scope of questions that we have been receiving from other people. So I'll come to that, but now first I'll introduce some topics in between the questions. So this seminar is not an introductory seminar. so if you are not familiar with the Red List, you should definitely take a look at the Red List guidelines, and the link is here, and also take the Red List online training course. I'm going to assume that most of you have already done this. So I'll go into more details and more difficult questions. But I do want to start with a few really fundamental concepts and topics. And the most fundamental concept really is what the point of Red List is. Why do we have the Red List? So the purpose of the Red List, as it is described in the Red List categories and criteria, is classification of species according to their extinction risk. And you all know this, but still it's important to repeat, because a lot of the misunderstandings about Red List actually originate from not really understanding the purpose of the Red List. So the purpose is not to list rare species or depleted or declining species or species that need protection. It's not to create maps for conservation. It's not to determine conservation priorities, even though, of course, Red List contributes to all of these. And as described in the very beginning of the Red List categories and criteria, the Red List may focus attention on those taxa at the highest risk of extinction, but it is not the sole means of setting priorities for conservation measures. Related to this, let's start with one of the questions. So this is a similar, we get similar questions to this quite often. So based on the available data, a species is assessed as least concern. But we know that there is overexploitation and illegal killing or wildlife trade. How can we assess such a species more accurately in order to save it? So first, starting with the more accurately part, there is no more accurately. So if the data are sufficient and the criteria have been applied correctly, then the least concern, whatever the result is from the assessment, is the accurate category. There is no more accurate assessing of that. So in other words, the effects of overexploitation, illegal trade, illegal killing and trade should already be incorporated. Although I'll go back to this topic on the next slide. In general, the Red List can incorporate the effects of all threats, even if you don't know what the threatening factor is. Now, coming back to this last part of this question, in order to save it, yes, of course, our ultimate goal is to save species, but the only way the Red List can contribute to that goal is if it is credible, and credibility requires consistent and objective assessment of extinction risk. So that's the most important thing that we focus on in developing guidelines and in applying the criteria. And in the standards committee, we try to ensure consistent and objective assessment of extinction risk according to these criteria. So I mentioned that the effects of threats should already be incorporated, and that's an important topic. One of the strengths of the IUCN Red List is that it's mostly based on symptoms. So in this next slide, which is kind of complicated, what I'm trying to show here is the types of information that are used in the IUCN criteria. You all know them. We use information on all of these, but I kind of put them into three categories. So the first one is what I call symptoms. These are the data on the particular population at the time that the assessment is made. What has been the trend of the population? What's the current range area? Is the population fluctuating? Are there subdivisions? All of these kind of information on the population of the species is the basic part of the assessment. Now, we interpret these data in the context of the species-specific traits. So the same trend means different things when observed for different species, depending on the generation length of the species. And similarly, species-specific traits on dispersal characteristics and abilities of the species, habitat preferences, breeding system, life cycle, etc., contribute to our interpretation of the data. For instance, to interpret population size, the number of mature individuals, we have guidelines that focus on the breeding system and life cycle and social structure. All of these species-specific characteristics, life history traits, are used to interpret the symptoms. Finally, we have threats. Now, to conserve species, it's of course important to know what the threatening factors are. But one of the strengths of the IUCN criteria is that even if you don't know what the threatening factors are, you can still do a red list assessment. It's not, of course, as much information. It's better if you know the threats, but even if you don't know the threats, if a population is declining at a certain rate, regardless of why it is declining and regardless of whether you know why it is declining, that species can be assessed as a threatened species. However, if information on threats is available, it can be used in the criteria, specifically and most commonly in projection of future reductions under criterion A3 and A4, and in defining location. As you know, location is defined based on threats, so we use the threat information for that. Okay, now a kind of related question. Data on a species indicates that the species should be listed as vulnerable. But this is a top predator, and there are studies published in the scientific literature that indicate that top predators are at a higher risk of extinction. So because of that, should this species be listed as endangered instead of vulnerable? You might have already guessed that the answer is no, definitely not. And why not? Because first of all, top predators, why do top predators have a high risk of extinction? It's because they have low population densities and long generation lengths, but these factors are already included in the red list. So if the data are sufficient and the criteria are applied correctly, vulnerable in this case is the correct criteria, and there's no need for any adjustments based on other factors. Now, one thing that's very common in all assessments and in all of science actually is that the data we have is always uncertain. Even for the best studied species, we cannot know, for instance, the precise number of mature individuals. So, and IUCN Red List is designed to take that uncertainty into account, and doing so really strengthens our assessments and strengthens the credibility of the red list. So always important to remember that all data are uncertain. So how do we deal with this? The most important thing is that you do not enter the information into SIS as a single number. You enter as an interval. For instance, you can enter the area of occupancy instead of a single number as a range of numbers separated by a dash. That means it can the area of occupancy can be, in this example, between 900 and 1,000 square kilometers. Now, in addition to that, you can also specify a best estimate. So in the second example, what the assessor has said is that the best estimate, the most likely value for area of occupancy is 700 square kilometers, but 500 to 1,000 square kilometers are also plausible values. Now, finally, you can also enter the best estimate as a range. So in the third example, the assessor is saying that most likely the area of occupancy is between 1,600 and 1,800 square kilometers, but it could be as low as 1,500, and it could be it could plausibly be as high as 2,300 square kilometers. So once you enter the data in any one of these formats, then the SIS criteria calculator will incorporate that uncertainty, propagate through the assessment, and as a result, the end result may be a range of categories. So you may say the species is listed as critically endangered, but plausible categories include both critically endangered and endangered. So IUCN Red List allows that, and the SIS does that calculation for you. Now, there are, of course, many different types of and levels of uncertainty, and there's a lot of guidance in the Red List guidelines about how to deal with them. In some cases, the answers are quite difficult and complex. So this is one of the difficult questions. How to properly extrapolate data from one group of species to another without generating uncertainty. Now, so in this example, the one group of species with little data or with more data is frogs, and the other is freshwater crabs that have similar habitat requirements. Now, these are completely different taxonomic groups, right? One is invertebrate, one is invertebrate. But if they are found in the same area and have similar habitat requirements, then they share these habitat requirements. But in that case, you can really only extrapolate decline in habitat quality, right? If the water quality is declining because of pollution, for instance, then it is likely that it will affect both groups of species. But you cannot really extrapolate anything else. You cannot extrapolate population sizes, population trends, area of occupancy, none of those, because those are all going to be species specific. Now, if you are using data from species not only with similar habitat requirements, but also similar taxonomy, body size, life history, etc., then you can do more extrapolation. Now, there are examples of these in different sections of the guidelines, and later in this webinar, I'll talk about generation length and how different studies have used information on similar species to help estimate generation length. So there are examples of these, but it's very important that to be careful with this, of course, that not to extrapolate any information that is not common to both species. Now, also something about the last part of this question, without generating uncertainty. This always generates uncertainty. There's no such thing as extrapolating and not having additional uncertainty. But the important thing is that don't be afraid of uncertainty. Instead, use it wisely. In other words, as I mentioned before, specify uncertainty carefully. And of course, if you are extrapolating between given species, you know that the habitat quality is declining for one group of freshwater species, then you are extrapolating to the other. Of course, the uncertainty will increase. Here's another difficult question. Again, this has to do with uncertainty and lack of data. How to assess widespread species with few and very small populations? So this is a difficult question, so I'll answer in a very general way for all these kinds of questions and a more specific way for this particular situation. So the general answer is always enter all data to SIS, even those data that you don't think will contribute to the listing. In other words, don't decide before you do the assessment which criteria should apply. Because that's not how the IUCN criteria are structured. They are really designed to, if you go through this red booklet, right, they are designed to assess under A and then B and then C and then E. The only time you stop is when one or more of those criteria are met at a given threat category. So you do all of the A, B, C, D, E for critically endangered. If none of them are met, then you do again all of them for endangered, and then see how many of them are met, right? So you don't decide beforehand what criteria is applicable. And again, as I mentioned, specify all data with uncertainties, use the SIS calculator, and do not predetermine which criteria to apply. Now, coming back to this specific question, widespread species, few and very small populations, in this particular case, the important information that will be necessary to really estimate carefully would include area of occupancy, locations if threats are identified, severe fragmentation, which is an important topic, and I'll spend a lot of time on severe fragmentation in the next segment, very restricted and number of mature individuals. But it's also important to consider all other data, especially related to declines and reductions. I'll talk about declines and reductions later too. So why is this widespread species, why does it have few and very small populations? Did it always have very small and few populations? If not, when did it decline? Has it declined in the last 3 generations? All of these are important considerations in trying to assess these kind of cases. Okay, now next I'll talk about severely fragmented, but I want to just take a moment to see if there are any questions. Anyone wants to ask any questions about the first part of the of my talk?
There are no questions posted at the moment, Resit. We've got a quiet audience right now, so I'm sure there'll be more questions later.
Well, I hope people are not sleeping. I am not sleeping, but I cannot promise that will continue. If people don't ask questions, who knows?
Oh, hang on, we have just received one question. What is the difference between inferred and suspected in criterion A?
That's a good question, and we have been getting that question quite a lot. So in the last version of the guidelines, we actually came up with a lot of examples to differentiate inferred versus suspected. And this is especially important, not for criterion A, but especially for other criteria. And the reason it's not as important for criterion A is that in criterion A, either suspected or inferred allows a particular decline rate to meet a threshold. In other words, for instance, in criterion C, suspected declines are not allowed, but in criterion A they are allowed. So and the main difference is the degree to which you have particular evidence. So inferred is inferred from specific information such as decline, specific measured decline in area of habitat, right? So that you say, okay, there is 30% decline in habitat. I am assuming that because it is declining, especially in the core parts of the range of the species, the population probably declined by more than 30%, right? So that is, that I would call that inferred. Suspected is more circumstantial. For instance, there is a substantial increase in the illegal hunting of a species concurrent with increasing efforts of illegal hunting. In other words, effort is increasing. People are spending a lot more effort to catch the species, and especially if then the number that is being caught is not changing much. That means despite increasing effort, they can only catch so much. That means the population is actually declining. So that might be suspected decline. However, if you have population-wide data for that, catch per unit effort and you calculate it based on actual calculation of catches and effort numerically, then it becomes estimated or inferred. So it depends on the how much data you have and how reliable the data are and how remotely related the data are to the population size. So take a look at the new version of the guidelines. There are a bunch of examples there. And if that doesn't clarify, then send us the question with as much detail as possible about what you actually know, what kind of information you have, the scope of the data, what spatial area that the data is coming from, all those details, and then we'll discuss that. Okay.
Thanks, Resit.
Continuing with severe fragmentation. Now, severely fragmented is defined as most individuals being in small and relatively isolated populations, right? So the first thing to note is that in IUCN criteria, severe fragmentation is not fragmentation of habitat, it's fragmentation of population. However, in many cases, we infer the fragmentation of population from the fragmentation of the habitat. Now, what does small mean and what does isolated mean? Small means that the population size is so small that there's a increased probability of extirpation. In other words, population is not viable in the long term. And isolated means that if this population is extirpated, there's a reduced probability of recolonization. So let's look at in detail what these mean. First, what is small? Again, there is in the expanded section in the guidelines. In the last version, we added a few paragraphs on this. So in the IUCN criteria, it says that small populations may go extinct. So the way we interpret what this means is that the size of the population alone is the cause of the increased risk of extinction. In other words, even if the population is not declining, the population size is so small that it is not viable in the long term. So that is how we define small. Just as a side note, some people ask if the concept of minimum viable population is applicable in this case. No, it is not. MVP is not applicable here. Don't spend any time on that. Now, what does it actually mean in practical terms? It depends on the species, so I cannot really make a generalization, and we don't really say any numbers in the guidelines. But we do say a few things that might help. First of all, I think that its population size in the low tens can be considered too small for most species, right? If there are only 10 or 20 individuals or 30 individuals even, I would consider that for most species, that number is probably too small, and I would consider that a small population for the purposes of assessing severe fragmentation. And again, for most species, a population size in the hundreds is probably large enough. Even if the area is small, if there are several hundred individuals, it's most likely large enough. So it's in between those two numbers. The threshold between small and not small for the purposes of severely fragmented is, I think, in between, so between 50 and 100 individuals, somewhere in there, maybe 200, maybe 40, some number like that. Now, how do you decide for a given species? So the type of evidence you use can, for instance, include the history of the population. So if there is a population that has, let's say, 40 individuals and that has persisted for a long time with that number of individuals, or there is a population that is occupying 5 hectares and it has been a similar density in that area for a long time, those type of data are an indication that those thresholds can be used to separate small from not small. So basically, history of persistence is very important. The other thing is, suppose you don't know the history, but you go to a place, and if there is continuing and sufficient recruitment in a population, so you are studying a plant and there are only 20 individuals, let's say, but you always see seedlings, they're growing, they're becoming mature. So there's a good age structure, there are all ages are represented. In that case, you can assume that number of individuals is sufficient to maintain that population. Now, coming to what is isolated. Isolated basically means too far from other populations for recolonization to occur with a high likelihood. So in this case, the type of evidence you use would be about dispersal distances of the species, dispersal ability of the species. And if you don't have any of this, dispersal ability of similar species. And by similar, I mean they have to be quite similar. It has to be same taxonomic group, similar body size, obviously the same mode of locomotion or motility. And based on those, you can make extrapolations and say, I'm going to assume that any two fragments that are, let's say, more than a kilometer away are just too far, and I will consider those to be isolated, right? So basically, you can, based on the biology of the species, you can come up with these thresholds and say anything that's more than, X number of meters is too isolated, and any population that's fewer than Z number of individuals or Y hectares is too small, and then interpret the distribution based on those. So let me give a few examples. So here's a case, and someone mapped the distribution of the populations, and the areas are proportional, let's assume, to the number of individuals, so the density is kind of uniform. And let's, for the time being, assume that all of these are far away from each other to be isolated. So in that case, would we consider this population to be severely fragmented? Well, with this information, of course, we cannot know, because we don't know anything about the actual potential number of individuals. Now, we know the area, but we need to have some very crude, even very rough idea of the number of individuals to be able to say if this is too small or too large. For instance, maybe even the smallest patch, let's say this one, has 2,000 individuals because it's an insect and these are forest patches, and this has 20,000 individuals, and etc. So in that case, it will not be considered severely fragmented, regardless of how small the patches are and how isolated they are. Now, let's put some numbers. Let's assume that these are the numbers approximately, based on the density. That's what we guessed. This is the number of individuals in these patches. Now, would you consider this to be severely fragmented? Again, even if we assume they are all isolated, we still cannot know, because we haven't yet decided what threshold to use for what a small population is. So if we say anything less than 40 is not viable, and anything 40 or more is viable, then this is not severely fragmented. Why? Because the number of individuals in the populations that are above that threshold is 140, whereas the ones that are below that threshold is much fewer. So most individuals are in larger populations. So it's not severely fragmented. To be severely fragmented, more than half of the individuals must be in small and isolated populations, right? So if you say, 50 individuals is my threshold, that calculation will change, right? Then if you say for it to be viable or for it to be not small, it needs to have at least 50 individuals, then this population is small, and these are all small populations. Now, majority of the individuals are in small populations, because if you add these numbers, you get a number 110, that's more than 100. So that will mean that this is a severely fragmented population. Of course, this, as I said in the beginning, assumes that these are all isolated. But if you, for instance, decide that this is actually close enough that these two populations are not isolated from each other, then again, this will not be a severely fragmented, because the number of individuals in the small and isolated populations is much smaller than the number that is in the other populations, in not small or not isolated populations. Okay, I hope that clarified how this idea is applied. Now, if you have more kind of details or other points to make, so as I said, it's based on subpopulations, not habitat patches, or especially not on locations. Now, a question that we got recently is can a taxon with a single subpopulation be considered to be severely fragmented? Now, yes, if that single subpopulation is small based on the viability considerations that you have decided. So if there is a single subpopulation and that population is too small to be viable, then it is severely fragmented. But this is a very rare case, rare in the sense that if there is only a single subpopulation and that is too small, then in that case, the taxon would probably meet criterion D at a higher threat level. On the other hand, if the single population is large, then of course it's not severely fragmented. Now, a taxon with two subpopulations can be severely fragmented only if they are isolated from each other, if the two subpopulations are far apart, and both are too small to be viable. But if the larger of the two is large enough to be viable, then obviously more than half the individuals are in the larger population, so it will not be severely fragmented regardless of isolation. And finally, sometimes there's a confusion with the concept of location, but these two concepts are actually quite independent of each other. A taxon may be severely fragmented, yet all the isolated subpopulations may be threatened by the same major factor, in which case there will be a single location, or it may be severely fragmented and each fragment or each subpopulation may be threatened by a different factor. In that case, there will be many locations, right? Because location is dependent on the distribution of threats to subpopulations. Another, of course, difference is that severe fragmentation does not require an ongoing threat. You don't need to know why the taxon is severely fragmented, or even if it was, a human impact or not. It doesn't matter. If it fits the definition, regardless of why it became severely fragmented, then it is severely fragmented. Okay, so that is all I want to say on that topic, and I'll next talk about reductions, unless there are some questions. Okay, so there are no questions at the moment. I found that a very clear explanation myself. Thank you, Caroline. It was very clear. It's good, especially the point about can a population with one subpopulation be severely fragmented or not? And I often get that question in training workshops. It's really good to hear the answer to that. Oh, someone is asking what the definition of a location is, but I think you might be covering that later. No, actually, I will not be covering that in this webinar. Please check the guidelines. The definition of location has to do with threats, but maybe in a upcoming webinar we can have different topics, and maybe we should focus on location because it's also a complex topic.
Yeah, that would be wonderful. Thank you.
Here's another difficult question. If recent field surveys did not find a species in the few localities where it was previously recorded, is this enough to determine a population reduction? It's difficult because we need to know a few other things. First, I would say that you need to carefully consider the difference between the three types of declines that I'll talk in a minute. But what we need to know in cases like this is, what areas were surveyed? Did they only survey the areas where the species has been recorded before, but were they any surveys, even if passive surveys, in other areas that the species that within the region? Now, passive survey means that the intent of the survey was not really to find that species, but for another reason, someone who could identify the species if it existed visited an area, right? So if someone is collecting, plants of a particular species for scientific research, but if they saw the target species, they could actually recognize that. And because it's a rare species, that is called passive survey. So we need to know those kind of things. We need to know something about dispersal ability of the species. We need to know about crypsis. In other words, how difficult an individual of the species is to see in different times of the year, in different times of the year, and in general the survey effort. So depending on these kind of considerations, a species like this can be assessed as suspected reduction or inferred continuing decline, but it can even be considered possibly extinct, depending on the survey effort and how long it has been since it was last detected. And there are new guidelines on how to determine if a species might be considered possibly extinct. There is a spreadsheet that you can use, and there are quantitative methods that are implemented in an easy-to-use way. But if you want help with that, we can also help with running that calculation. Another related question, another difficult question about declines and reductions. While population of a plant species in commercial use may be getting smaller, there is fairly reliable anecdotal evidence, such as increasingly longer time to collect a given quantity, or the species disappearing from localities where it used to be abundant in the past. However, these are not focused surveys. Should this species be assessed as data deficient? I would say that given all this information, probably not. The species should not be considered data deficient. Again, it's important to consider three different types of declines that I'll talk about. But I would say that it's probably not data deficient because criterion A2 can be used with suspected reduction. And there are, as I mentioned, there are new guidance on that in the new version of the guidelines document. And continuing decline can be inferred based on this information. So criterion B or C can be used. But also if there is any trade information, population sizes can usually be at least approximately estimated. Methods of like catch per unit methods and similar methods can be used to approximately estimate the population size, even if it's very uncertain. We don't know whether it is 500 individuals or 5,000 individuals. That is actually, even though it sounds too uncertain to be of any use, it is still useful. Why? Because it is still below the threshold for number of mature individuals under criterion C, right? So even very wide range of uncertainty may actually allow assessment in some cases. Now, what are the different types of declines? First of all, there is the continuing decline and reduction. Reduction is under criterion A, and continuing decline is under criteria B or C. Reduction is quite different from continuing declines in several respects. One is, as I mentioned before, it can be suspected or inferred as well as observed, estimated, or projected. And the other more really important difference is that there are no additional conditions to use the reduction. In other words, if the population is declining by a certain percentage over a certain period of time, which is 3 generations or 10 years, regardless of all other attributes like population size or fragmentation or area or anything else, the species can be listed. Whereas continuing decline requires additional conditions for the species to be listed. And there are 2 types of continuing decline. Under criterion B and C2, we have continuing decline at any rate, meaning that this decline is expressed only as yes or no. It's true or false. It's either there is continuing decline or there is no continuing decline. Now, of course, as with any information that goes into the red list, that can be uncertain. So you can say if true is 1 and false is 0, you can say, well, it's probably, but I'm not so sure. So it's between 1.0 and 0.8, right? So that's uncertainty, but that uncertainty can be incorporated and specified. But the important point is that the units is Boolean. It's either there is decline or not, which can be uncertain. Whereas the units for estimated continuing decline under C1 is percentage, just like reduction. But unlike reduction, it is different amounts of decline over different time periods, right? For under C1, we have for endangered under 1 generation, critically endangered under 1 generation, endangered under over 2 generations, and vulnerable over 3 generations. So the periods is different, and the amounts are different as well. And as I mentioned, for both estimated continuing decline and continuing decline at any rate, there are additional conditions. For estimated continuing decline, it's only number of mature individuals under C1, but for continuing decline, it is range, area, locations, fragmentation, various other conditions are necessary for the species to be threatened based on continuing decline. Now, reduction and decline are complex topics, and they are covered in several different sections of the red list, and these sections are updated quite frequently. So I encourage you to consult the guidelines and consider if the information you have can support any of these 3 different types of declines and reductions. Now, one important thing that I want to mention is that declines, when they are expressed as percentage, are measured over generations or number of years. So the point of this is to scale the declines with respect to the life history of the species. So for that, we use generation length, and that's a very important aspect of the Red List criteria. It's defined as the average age of parents of the current cohort, and I'll go into the details of what this means in a moment. Generation length is greater than the age of first reproduction or age of maturity, unless the species breeds only once. obvious if a species only breeds once and dies, like salmonids and other species like that, then the age of first reproduction is also the age of last reproduction, which is the lifespan, and also the generation length. They're all the same number. But if there is a possibility that some individuals can breed more than once, then the generation length will be greater than the age of first reproduction, and it will be less than the reproductive lifespan, the age of the oldest breeding individual. So it will be between those two numbers. And generation length reflects the turnover rate of breeding individuals in the population. And we use it in the Red List criteria to scale all time-based measurements to account for the different rates at which different species reproduce and survive. And another point to make is that we always use the natural generation length, not the generation length that is changed because of a threatening process. Specifically, a lot of different forms of harvest reduces the generation length of the species, and we use the natural generation length. Okay, now what are the different definitions? So first I will go over the formal definitions and what they mean, so you get a sense of what we are trying to measure, and then I will describe two simple methods of calculating generation length. So the first definition is the average age of parents of the current cohort. So you basically look at all the individuals that are born this year, and then record the ages of those of the parents of those individuals, right? So if you made a histogram of that, you'll get something like this: the age of the parents of the current cohort. Some newborns will have really young parents, and some newborns will have really old parents. And the average of this distribution is the generation length, is one definition of the generation length of the species. Another definition is mean age at which a cohort of newborns produce offspring. So what does this mean? You take a cohort, meaning you look at all the individuals that are born this year, and then follow only those individuals throughout their lifetime, right? So some of these individuals that you start following will start breeding at a young age, and then the number of individuals of that cohort who are actual breeders will increase, and then some will start dying, so that will decrease, and then there will be an age of last breeding. Again, the average of these ages is the generation length of another definition of the generation length of the species. And the third definition is age at which 50% of total reproductive output is achieved. So in this case, imagine that you are following a single individual throughout its lifetime, a typical individual, whatever that is, and that individual, starts breeding at some age, and usually breeds not as many offspring. For instance, if it's a plant, there will be only a few flowers at first, only a few seeds, and then it will increase, and then sometimes there will be senescence. It doesn't have to be this way, of course, and then there will be an age of last breeding, and then the age at which 50% of this total reproduction occurs is the generation length. Now, these three formal definitions, actually, if you have lots and lots of demographic data, you can calculate all three of them from, if you have a complete life table, you can calculate all three of these, and they will be somewhat different from each other, not very different, just slightly different, except for some strange life histories, the difference may be a bit more. But for the Red List, any one of them is acceptable, and it will be extremely rare that you actually have data to calculate more than one of them. Okay, so those are the conceptually what generation length is about. Now let's look at some practical examples of how we calculate it. So in many cases, we only know the first and last ages of breeding. Right? We know the the age at which the individuals reach maturity, and then we kind of know the lifespan. Sorry, we know the lifespan, and then we can calculate generation length based on this formula. So you calculate the length of the reproductive period. In other words, the difference between last age minus first age. That's the length of the reproductive period. I'm sorry, I don't know what's happening here. You multiply it with Z, I'll come to that, and then add the age of first reproduction. Now, Z is a number between 0 and 1, and it's usually less than 0.5. For mammals, there is a paper that calculated the Z to be around 0.29. And there's another paper, we calculated Z for birds to be between 0.14 and 0.32. And you can look at that paper and find which one fits your species better. Z can be estimated from related species. So it's okay to use an approximate number based on related species. So you can even use it, for all mammals, you can use 0.3. That's fine. If you have more information, of course, the better. If you know the Z for, the order of mammals for which you are calculating, then you can use that. That's better. So here's an example. Species age of first reproduction is 3 years. Last reproduction is 10 years. Z is estimated to be 0.3 based on species in the same class. So the generation length will be age of first reproduction, 3, plus Z, 0.3, times the length of the reproductive period, which is 7 years, from age 3 to age 10. So that will be 3 plus 2.1, it will be 5.1 years. So that's an example of using this information. Here's another way of calculating. Sometimes we don't know the age of last reproduction, but we know adult mortality. Then we can use this formula, which is 1 over adult mortality plus age of first reproduction. And this is especially useful if annual mortality after the age of first reproduction does not change much and is well known. If mortality and fecundity do not change with age much after the first age of reproduction, in other words, there is no senescence, then this is a good formula to use. And here's an example. Age of first reproduction is 2 years. Annual adult survival rate is 60%, which means that mortality is 40%. So 1 over mortality, 1 over 0.4 is 2.5, plus age of first reproduction, which is 2, generation length will be 4.5 years. Okay, so here's a question that uses these concepts. The question is, I have the following information for my species. Age of first breeding is 3 years, last breeding is 15 years, annual adult mortality is 0.66, and Z is thought to be between 0.3 and 0.4. Now, based on the second formula I showed, generation length will be age of first reproduction plus 1 over mortality, so that will be 4.5 years. But if you use this information on Z and the age of last breeding, then it will be 12 years of reproductive period multiplied by 0.3 or 0.4, added to 3, which is the age of first reproduction. It will give somewhere between 7 or 8 years. So the question is, which one is correct? Is it 4 or 5 years or 7 or 8 years? These are quite different numbers. So what's the answer? This is actually a question that I ask my ecology students sometimes to see if they have learned the concepts of survival and reproduction correctly. So to get to the answer, first you have to really carefully consider what these data show? So it says annual mortality is 0.66, which means that annual survival is, adult survival is 34%. In other words, every year, only 34% of adults survive to the next year, right? Now, we don't know what the juvenile survival rate is. Usually, juvenile survival rate is even lower than adult survival, but let's assume that it's the same. In other words, juvenile mortality is the same adult mortality. So that means every year, only 34% of individuals go to the next, survive to the next year, right? So if that is the case, how many individuals do you get left after 15 years? So that is calculated by multiplying 0.34 15 times by itself, because every year 34% survive. So you multiply 34%, 15 times. That gives you a number that is about 10 to the minus 7. So about 1 in a million. Okay, so which suggests that there is something wrong with this data, right? Because this says that in order to be able to observe an individual breeding at 15 years, if these data are correct, you have to be either extremely lucky or observe at least 10 million individuals, right? And know their ages. This is very, very unlikely for a threatened species. So there is something inconsistent. The data on the last age of breeding and annual mortality are inconsistent, meaning that one or the other is overestimated, right? Age of last breeding may be overestimated because maybe that number is 15 years is coming from captivity. It doesn't apply in natural environments. Or maybe annual mortality is overestimated because it's coming from a study in which dispersal outside of the study area was not considered. So it's actually not actual mortality, it's apparent mortality, right? So it's important to really carefully check the original data and be careful of what, how reliable the data are and where the data are coming from. Okay, here's another difficult question. How to calculate generation length for a species with a persistent soil seed bank if seed viability and residence times in the soil seed banks are unknown? Let me first explain for those of you who are not plant biologists. Residence time is time to germination. That's the time that the seed spends in the seed bank from the time that it's deposited on the soil to the time that it germinates. And usually this excludes seeds that never germinate, right? So now, in general, how do we calculate generation length for a plant with or without seed bank? So if it is possible to age mature plants, so for instance, if these are trees, you can age them, count the rings, core them, count the rings. So what you do is you take a random sample of all seed-producing plants, all mature plants, determine their ages, and calculate the average of that age. That will be one estimate of generation length. However, and here is the difficult part comes, this is like to be years since germination, because you can age the mature plants. They put rings every year after they start growing. You don't know how long time they spend in the soil as seed. So this is years since germination. If it may underestimate years since the individual was a seed, right? So what can you do? Well, what you do is approximate. So you can add the median and not maximum seed residence time of related species in the soil. So suppose that you calculated this average. You say mature trees on average are 30 years old. We don't know how long the seeds have stayed in the soil, but in related species, people have observed that the seeds germinate within the 5 years or they die, they don't germinate. So maximum residence time in the soil is 5 years, and on average it's probably 2 or 3 years, right? So, and then you can add that to the 30-year average age since germination, and then you'll get a more approximate, a more accurate estimation of the generation length. Now, it's really important to go through these thought exercises and calculations, because it's better to use an approximation than not use generation length at all. Why? Because if you don't use generation length, that means you cannot use criterion A, you cannot use criterion C1, and you are really leaving a lot of information out. You cannot use reduction criteria, which is really important. So it's important to use an approximate, even if it is uncertain, even if it is potentially underestimating or overestimating. It's okay. It's okay to use it. More important to make sure that generation length is considered. Okay. Questions?
There are no questions in the Q&A at the moment. Thanks very much for covering generation length, Resit. There are a few questions coming into the chat about how to calculate those Z constant values. I have already posted where to find the section on generation length in the Red List guidelines, so it's section 4.4, and in there are a couple of references that about how to calculate how the Z values for mammals were calculated. So it's probably useful that people look at those references to see how they approach that challenge, and see how that would relate to your own species as well. And Resit just posted the link to the birds calculation.
So yeah, I know the bird. I don't. The other one is as Caroline mentioned, it is referred to in the generation length section of the guidelines for mammals. Yeah. But also, I mean, if any of you are interested in doing similar calculation for other groups of species, I think there is definitely room to do similar calculations for other groups of species. For birds, it was between 0.14 and 0.32, I believe. So yeah, 0.15 to 0.30, let's say, approximately.
Yeah, that's right. What I tend to find often in assessments coming to the Red List unit is that people assume a Z value of 0.5 because they don't have the information at all. Would you say that is an appropriate thing to do, and just base that as a maximum generation length for the assessment without the additional information about what Z actually realistically can be?
Well, what I would do in that case is that I would also use a much lower value. Mm-hmm. So if it's a group that is not at all related to birds and mammals, I would say use two very different values, 0.2 and 0.5, let's say, okay? And calculate the generation length with both, and then calculate the reduction with those two different generation lengths. And that gives you the total uncertainty in the reduction because you calculated over two different generation lengths. In many cases, that won't make much of a difference, because especially for long-lived species, most of the reduction would be more recent. It won't be 3 generations ago. It would be like the last one or two generations. So the 3 generation length, whether it is 10, 20, 30% more or less, it won't make much of a difference. But for some species, it would make a difference. So for those, it would be important to know how much difference it's making. Just use two different numbers, get two different reductions, and enter both as plausible values of reduction in population size. I think that's the right thing to do in those cases.
Brilliant. Thanks, Resit. And a good reminder that uncertainty is key to document in assessments here. Thank you.
Thanks. Okay, so we still have a number of minutes, so I'll talk about climate change a bit. This is a very, actually, of course, difficult topic. It's complicated, and there are developments all the time in the literature, and we try to follow those and reflect those in as much as possible in the guidelines. So how do we incorporate climate change related threats in the red list? First, just a note that the part of the guidelines in which this is discussed is section 12.1, and the main part of that section is the step-by-step approach of using different types of information and getting more and more complicated to assess a species. Now, as I mentioned before at the beginning of my talk, the Red List criteria are mostly based on symptoms of endangerment, such as population size, small area, small population size, which means that in most cases, it's not necessary, although of course it's always helpful, to understand why a species is declining, to understand the mechanism of declines, or even to know what the threats are. So because of that, Red List allows incorporating indirect effects of human responses to climate change, as well as direct effects of climate change. What I mean by this is this. A lot of the species are threatened by climate directly because their current range is becoming inhospitable. But in some cases, they are actually being threatened indirectly because of climate change. Humans are shifting where they do agriculture. Agricultural lands are shifting because of climate change, and that shift is causing species to be threatened. If we have good enough data on species, area and range and reduction, both of these mechanisms would be incorporated in our assessments. There is also, of course, the possibility that for the species you are assessing, climate change may be on your mind, but other threats may be more urgent. If a species is being poached, at a high rate, it might go extinct before climate has any effect of that. In any case, so when climate change information became more and more common, we were asked if Red List can actually assess species that are threatened by climate change. And we said, well, we don't know. When Red List was developed, we didn't know anything about climate change when the criteria were first developed in the 1990s. So let's see, let's test it. So what we did is that we did a study. I won't go into the details, but what we showed is that if there are sufficient data to use at least two criteria, A and B, or B and C, or A and D, if there's sufficient information to use at least two of the criteria, then the Red List gives enough warning time for species. So the time between when a species is listed as threatened to the time that it would go because of climate change, we call that warning time, and that is quite long for most species. Only for 1% of that of those species, it was less than 1%. But if if you wait until the species is critically endangered, more than 50% of species may go extinct within 20 years. So how quickly conservation action is taken makes a big difference. But anyway, so based on that, what we decide is that the first option, which is not what we want to do, but it's possible to do, is to make the red list assessment as before without explicitly considering climate change. This would still give us, in many cases of species being threatened by climate change, even if we didn't know it was being threatened by climate change, it would still give us good information, an accurate assessment. But it may not be sufficient for some species, right? But it's better than anything else unless you have more data. So the Red List criteria actually is the only climate change vulnerability assessment system that has been quantitatively tested and validated. So that's important to know. So just because climate change is not specifically mentioned in the categories and criteria doesn't mean that it's not actually detecting species declining because of climate change. However, even if you go with this option, it's very important to use as many criteria as possible, follow the guidelines, and use appropriate time windows, just as described in the guidelines. Even then, you have to keep in mind that the assessment may underestimate extinction risk due to climate change. So what else can you do? Option 2 is to do the same thing, but consider the mechanisms of the climate change impact. This is important because some of the key red list parameters, specifically these three, but especially the locations, can be defined based on climate change impacts. And that may actually change the assessment, right? Because if you consider climate change impact as a general threat, and the way that it's causing the specific mechanism by which it's threatening the particular species you are assessing, is spread spatially in such a way that there is a single location that will increase the threat level for your assessment. So that's there are details in the Red List guidelines in section 12. So even if you don't do any modeling, any specific climate change related projections, if you just keep climate change effects and the mechanism by which climate change is impacting your species, if you keep that in mind as you define key red list parameters, that will go a long way to incorporate climate change impacts in red list assessments. And the third option is a bit more complicated. It requires developing climate-based species distribution models. So species distribution models, also known as bioclimate models or ecological niche models, are basically statistical methods to predict the future potential habitat or distribution of a species based on current occurrences. And there are different methods of doing that. There are detailed guidelines for use in red list assessments. And based on those projections, you can infer reduction or continuing decline due to climate change. And that can be used in criterion A3, A4, B, or C. And that, as I mentioned, there are detailed guidance in the Red List guidelines. But the problem with this is that it requires data. It requires occurrence data, climate data, and land use data. And it requires a bit of modeling expertise. And there are actually, I'm happy to see that there are more and more groups actually doing these assessments. We just reviewed a set of assessments on South African plant groups that were done using this approach. So it's possible to do. It doesn't require that much understanding of, you don't have to do actual coding because there are tools for doing that. And there have been plans, but it's very difficult to actually do this. But we are planning to develop training materials and online tools to make this even easier. There is a fourth option, which I won't go into the details of because it requires even more complicated modeling. It requires also population models that is added on top of the ecological niche model. So basically doing a criterion E type of assessment. So this is also mentioned and described in the guidelines, but it requires a lot more data. So in summary, you can choose to make Red List assessment as before. Make sure to use as many criteria as possible and use the correct time windows. And the second thing you can do is to consider the mechanism by which the climate change is impacting your species when you decide how many locations there are, whether there are extreme fluctuations or severe fragmentation in the context of climate change. And the third one is to develop, and the third and fourth one are to develop models of different complexity to infer declines in the population size based on climate change projections. So that is all I have to say. If there are any questions, maybe we can actually unmute people and people can ask, or they can, but however way you want to handle it, Caroline.
Thanks, Resit. I'll ask Clay if he has the power to unmute people, because I don't have that power, but at the moment. So we do have one question that has just come in. I've not read it yet. So what if one criterion has extreme uncertainty and yields a listing of critically endangered, but another criterion is much more certain but yields a listing of endangered? Normally you would go with the more threatened category, but here that category is much more uncertain. So which one would you go with?
Very good question. So it depends on the details. I would like to see the actual data, but I would say the easiest way is that you enter all the information with the appropriate uncertainty into SIS and use the criteria calculator. So what that does is that it will, depending on how you specify the uncertainty, it will propagate both of those uncertainties, and it can, for instance, the one plausible result of that would be that it would be listed as endangered with critically endangered as a plausible category. But it will, to some extent, depend on the attitude settings. And there are, in SIS, these are actually fixed, and we have made a decision of where to set these attitude settings. Attitude settings are basically, as the next question is asking, precautionary versus evidentiary. How much evidence do you need before you say that a species is threatened, versus how precautionary you are, and do you consider a species threatened even if there is very little evidence? Now, in principle, you can have any of these attitudes, but for the global Red List assessments, we have actually made specific suggestions of where these should lie in this, and basically we suggest a precautionary but not extremely precautionary attitude. So basically, just slightly more precautionary than the very middle of the range between precautionary to evidentiary. So that's what we suggested. So I, as far as I know, actually I don't use SIS much, but as far as I know, that is actually fixed in SIS, and you can and when you use SIS criteria calculator, it uses that set of attitudes to make the assessment.
Thanks, Resit. It does indeed. There's another question which Clay is currently answering: how to cope with this species which is found only once. Now, there is guidance on this in the Red List guidelines. I think Clay is going to direct you to that section as well. Okay, well, I'll let Clay continue answering that question, and that's all we really have time for tonight, or today, tonight for me. So thanks very much, Resit. That is fantastic. As always, you've given great insight into the Red List criteria and answered all the difficult questions that are burning in people's minds. And as always, we really appreciate your time and sharing your knowledge on this. So thank you again.
You're very welcome.
Thanks to Albuquerque Biopark for helping making these webinars a reality. In particular, thanks to Clay Meredith for his very practical solutions to what I thought was a difficult and challenging task of reaching out to such a large and valuable network. And of course, thanks to all of